There we go. And before we actually get to Kristen, I just want to remind you that if you're seeing this video, you're watching the actual video, or you're seeing it in the future after it's been recorded. And if you're not a member of Lab, please consider joining Lab. Go to labirders.org and click the join, renew, or you can donate. Either one, any of them, we'd appreciate. All of them, we'd certainly appreciate. But that helps us. That helps support these webinars, our science projects, our scholarships, our and other things that go on at Lab. And with that, I would like to introduce Janet, who will introduce our speaker for tonight. Janet.
Great. Thanks, Ron. So Dr. Christen Bossu is a research scientist at Colorado State University. She received her PhD at Yale University in 2012, and since then has done postgraduate work in Sweden and in the US. Dr. Bossu is a member of the Bird Genoscape Project. Tonight, she will tell us how the project maps migration patterns in birds using genomics and how this information can be applied to the conservation of bird and other wildlife populations. The Bird Genoscape Project aims to map population-specific migratory routes of 100 species of migratory songbirds using genomic approaches. The BGP collects feathers at each stage of the migratory cycle. Professor Walt Sakai and his team of banders contribute tail feathers of misnetted birds to the project. The genetic information in the feathers is used to determine where it came from, making migratory connections across the annual cycle. These genomic results can be used to define important areas for migratory birds at finer spatial scales than previously possible. These areas can then inform conservation efforts and serve as the basis for understanding the impacts of climate change and other human-caused stressors on bird populations. So sit back and relax as Dr. Bossu reveals some fundamental insights into the genetic basis of species survival so that we can take action to protect them. So please welcome Dr. Bossu.
Thank you. I will start sharing my screen now. I want to thank LA Birders for inviting me, Mark and Ron for your bird jokes, and Janet for your introduction. Okay, so as Janet had mentioned, my name is Christen Bossu. I'm a research scientist with the Bird Genoscape Project. And the Bird Genoscape Project is a joint venture between two co-directors, Kristen Ruig at Colorado State University, which is where I'm based at, and Tom Smith at UCLA, which is actually where I started as a postdoc fellow with the Bird Genoscape Project. So tonight, I want to talk about basically what the Bird Genoscape Project is, our motivations underlying the development of this project. I want to introduce what a Bird Genoscape is and how we create it. And then the second half of the project or the presentation, I actually want to present some additional applications to conservation that we've done once we have this wealth of genetic data. So the title is New Insights into Migration and Conservation Through Bird Genoscapes and Beyond. I think the beyond is, bird genoscapes are the first step, and then we have data to ask so many questions. So I always start with this dramatic result that was published in Science in 2019. And the fact that 2.9 billion birds were gone since 1970. And I don't know about you, but this was shocking to me, and shocking and very sad. But one of the things that motivates us at the Bird Genoscape Project is a majority of these, the greatest impact are on migratory birds. So the Bird Genoscape primarily, but not exclusively, focuses on migratory birds, these neotropical migrants, where they breed in North America and then migrate south of the US border to the neotropics where they overwinter. So point in fact, they spend most of their time actually not in the US on their breeding grounds. And what this study had showed is we had lost 2.5 billion migratory birds since 1970, and there's about like a 28% population loss in migratory birds. What the authors did not do in this publication was look at causes for these declines, but in order to prevent future declines, we need to understand the drivers of such declines. One of the ways we do this, and we have to think about like where these stressors are occurring, and they actually occur across the entire migratory bird annual cycle. So on the breeding ground, along migratory routes, and on wintering grounds. And they range from habitat destruction, predation from animals such as household cats. This is an example of avian flu, but in one of the projects I've worked on, we see an increase in botulism at a migratory stopover in Great Lakes that is affecting common loons. So just avian diseases, we're seeing that as important stressors. And also just we're in a globally changing world with climate change and extreme weather patterns that can really affect the fitness and populations of these migratory birds. But we have to ask the question is, where are the birds most threatened? Is it on their breeding ground, on the migratory grounds or migratory stopover sites, or is it on their wintering grounds? And to this end, it's actually making these migratory connections that is the essential first step to conservation efforts. So if we see habitat destruction on the wintering ground of this yellow warbler in Southwest Mexico, what we want to do is we want to know which populations is it going to affect. And so it's actually the development of these population-specific migratory pathways where we can understand whether this habitat destruction on the wintering ground will have cascading effects on one breeding population or all of them, multiples. To this end, the Bird Genoscape Project, we use high-resolution genetic tags. So every time a feather is collected from a bird, thank you, Walter, tissue or blood at the base of the feather contains DNA. And we can use this DNA to trace that feather back to the breeding origin. What this means is that any feather and of any bird or carcass captured anywhere along the migratory trajectory can be traced back to where it was breeding. Today, I want to take you through an in-depth example using one species, the Willow flycatcher. And this is a really interesting bird because it has interesting conservation applications. For instance, as a whole, as a species, it is a species of least concern. It is doing pretty well, but not everywhere. So when we actually take into account subspecies boundaries and population trends, it is the Southwest willow flycatcher that is considered endangered. So a critical question for these declining, Southwest willow flycatchers is, what is the migratory pathway of the endangered Southwestern willow flycatcher? Where are they overwintering and where are they migrating? And we need to make these links in order to understand potentially where to focus conservation efforts. So to do this, the first thing we do is we scan the genome for genetic variants. So to put it simply, the genome is just all genetic variants, all the genetics that are within an organism. And this is just, in this image, it's just simple A, C, Gs, and Ts. This is your DNA. So what's interesting is when Christen, my advisor, started this in 2014, she did this sequencing method. It's called reduced representation sequencing. It's called RADseq for short, where it's basically you just sequence a fraction of the genome at high coverage. And she started doing this with 22 individuals, ended up with thousands of loci. But you can imagine 22 individuals across the genetic or the geographic range of the Wilson's warbler. It was amazing that she found the population structure she did, and she was able to make the inferences she was able to. But in this day and age and next gen sequencing, costs have dramatically decreased. So now we can actually do rad sequencing, or sequencing a fraction of the genome for like hundreds of individuals across the breeding range. Again, just to get the full geographic span. We can also, again, have thousands of loci, and we can also assemble a reference genome of the species we're looking at. And what this allows us to do is annotate the genome and actually look at specific genes, which is interesting. But like I said, technology changes, and what is more, and what we can do now is actually low coverage whole genome resequencing. And what is great about this project, or this sequencing platform, is now you're not just sequencing a fraction of the genome, you're sequencing the entire genome, everything. And we can do this for 200 to 300 breeding and wintering individuals. So the fact that we can actually do this with feathers is ideal. But what we end up with, like millions of loci. No matter the sequencing platform, what we aim to do is look for these unique genetic tags. We look for differences within the genome that represent the population structure of the species that we're looking at. So, and with these tags, what we do is we build a map of genetically distinct breeding populations. And this is, in fact, what a genoscape is. It's a spatially explicit map of the genetic variation across the breeding range. So for the Willow flycatcher, we've identified 7 distinct breeding clusters. We have this Pacific Northwest breeding cluster. We have an interior west distinct genetic breeding cluster, an eastern cluster, and the Southwest Willow flycatcher is its own distinct genetic cluster. We also identified 3 much smaller and unexpected ones, genetic clusters in the White Mountains, Kern, and Southern California. And so with these tags, we can use DNA from feathers to ID the breeding area of migrants. So I want to stress, and I can't stress this enough, this is an extensive citizen science-based feather collection. We partner with bird monitoring stations across the breeding grounds in order to collect migrants as they migrate through these stations. Tail feathers are collected and sent to us, where we catalog and store. And we also have this for wintering sites down in Mexico, Central America, and South America. So the Bird Genoscape actually houses approximately 250,000 feathers collected for about 100 species, and we span like a 25-year time series. And these numbers are only growing because we're always collecting for new projects and are what we consider bird genoscape species, although these numbers keep increasing as well. We keep adding species to the list. So this is a map of where we have the Willow flycatcher genoscape up above, where we have the breeding range. And what we want to do is we want to know where these genetic clusters are wintering and where they're migrating. And what we do is with these, with the DNA from feathers, and each circle here is going to be one individual, one feather, and we assign it back to the breeding range. And so what we found for at least the Pacific Northwest is they winter in Mexico down to Central America, and they primarily use the Pacific flyway when they're migrating through. The interior west breeding population, they winter from Central America down to Ecuador. And we found that they use both the Pacific flyway and the Central flyway. We have assigned migrating birds to those flyways. The eastern population, again, found shifted a little further south, so also found in Central America to Ecuador. But we find that they use the Central Flyway, Mississippi Flyway, and the Atlantic Flyway. But the question is, where does the endangered Southwest Willow flycatcher overwinter? And so we find that they are localized to Nicaragua and Costa Rica. And so this map here, so much goes into this, right? I mean, we have hundreds of birds on the breeding ground, thousands of migrants and wintering birds. And then we can find where, and this is the spatial context of where willow flycatchers are wintering. But every time a feather is collected, we also get a date. So we also have a temporal context. And so this is a video of what happens. I think many of you, I'm sure many of you use eBird, and you've seen the really cool projections of different species and how they migrate through, or you see that how abundances change. So you can basically see timing of migration of these species. What we do, and what this is what we are able to do is put this a population specific context on it. So let me start this, and you can just, so these are all the feathers that were collected, with their dates, and we can start to see in May is when we start seeing the Southwest Willow Flycatcher, and these are the breeding birds that were sampled. You can see in July, they're still Pacific Northwest migrating all the way up to the northernmost part of their range. They reached that in August, I believe. And then you start to see them disappear. And this is when they're starting to migrate down. And what you'll notice here is from October to November, there's really not a lot happening. And it's not that the birds aren't migrating, it's we just don't have feather samples from those locations. And then in December, you start to see the birds, or at least when we started collecting feathers down below. And in January is when we start to see the Southwest Willow Flycatcher occur on their wintering ground. And I think this should stop now. So this is the temporal context of what we were able to collect and identify with genetics. Oh, that's not what I wanted to do. So our goal is to map the migratory flyways of 100 species of concern by 2025. The number has always been 100, and since I've been with the Bird Genoscape Project for the past 4 years, it's been 2025, but that is, fast approaching. And every time I say this, I get like heart palpitations, and the fact that we still have a lot to do by 2025. But to this end, we have completed several genoscapes, and these are just some examples. We have a genoscape for common yellowthroat, Wilson's warbler, yellow warbler, common loon, willow flycatcher, and American kestrel. And what's great about this is these are the ones complete, but these are not the only species we're working on. We're currently working on over 20 species, and with collaborations or collaborators, we keep adding more to this list. So this is probably changing as we speak. And what I want to note is all of these genoscapes are in different stages of completion. Some of them are complete, some of them actually we were not able to create genoscapes for because there's no genetic variation within a species. And others are just beginning. We're just in the sampling stages. But what's great about these bird genoscapes is while they are critical and essential for conservation efforts, they're just a first step. For all of these species, we might have like population-specific migratory connections, but we can ask a lot of questions with the wealth of this genetic data that we do have. And for the second half of the talk, I do want to just talk about 3 of these main applications that we're really excited about. So the first one, it's an application 1 is to improve estimates of climate vulnerability. This is a way we can look at which populations are most vulnerable to climate change. And in that way, we can prioritize these species. We had a postdoc, Rachel Bay, with the Bird Genoscape Project. She is currently a professor at UC Davis. But what she did is she found that there were, with this data, what you can actually do is you can look at not just population structure, but you can look at what parts of the genome are associated with environmental predictors of the species. So the yellow warbler is highly driven by precipitation patterns. And what she found is that birds that are vulnerable to climate change are actually already declining, which suggests that what we're seeing now, the declines we're seeing now, we might not have to wait to the future to see, especially vulnerable populations. Climate change is happening now, and we're seeing signals within these population trends. Kristen Ruig also looked at these climate vulnerability and these and the environmental variables that underlie willow flycatcher climate vulnerability. And what she found is that the southwestern willow flycatcher was actually the most vulnerable to future climate change. And what she found is the southwest willow flycatcher in the future, that population has to make the most, the greatest molecular change in order to keep pace with the climate change, which is why this one is considered most vulnerable. A second application, which I'm excited to present here, is where we look at the genetic basis of migratory timing in American kestrels. The American kestrel is a diurnal raptor. And what we know in terms of migratory birds, timing and synchronizing migration with resource peaks has important fitness consequences. So we have a collaboration with Julie Heath from Boise State, who works on American kestrels and raptor research. And we also work with Barbara Helm, who we consider our circadian rhythm guru, our circannual timing. And what we know at least for birds, migrating birds, is that it's a complex interaction between biological clocks, so endogenous biological clocks. So inherited internal circadian clock and circannual clock. They tend to be synchronized by light input pathways like photoperiod, but there are additional environmental inputs that can in fact modulate or regulate the circadian clock, and that includes stress, nutrients via metabolic sensors, and temperature. So the project that was actually just recently accepted into publication, what we wanted to investigate was whether these core clock genes noted here, or whether less known genes that actually are involved in regulating these core clocks might be involved in migratory timing in American kestrels. So in a previous publication, we had shown that population structure of the American kestrel is related to migratory strategy. So this is the genoscape for the American kestrel. And what you can see is that in these northern ranges, these blue, yellow, and cyan, these are the migratory and partial migratory populations in the East, West, and Alaska. But what we also noticed, and what was very clear, is that Texas is a resident population and Florida is a resident population, and they were quite distinct in their genetic structure as well. So what we did is we compared the genomes of the residents to the partial migrants and the migratory birds. We identified top migration-linked candidate genes that have been found in vertebrates, from mice to other migratory birds. We created assays, these genetic tags, and actually genotyped all of these breeding birds for all of these circles. And then in Boise, Idaho, we had a migration station where we genotyped 165 birds across the autumn migration period. And what we found was quite amazing. 4 out of the 9 candidate genes that we had identified and that are known to regulate biological clocks is significantly correlated with autumn passage date. What we were able to identify is the existence of these migratory chronotypes. So these birds that have different allele frequencies, and these are just 2 gene examples, so top 1 and peak 1. So birds with this allele frequency, higher allele frequencies of top 1 and peak 1, these are the early migrants. So we can predict which birds are going to be traveling through. And then we have a late migration chronotype. And so it's actually genetic variation at these sites that suggests that American kestrels in the West have this adaptive capacity to respond to shifts, these phenological shifts in the face of climate change. Julie, our collaborator, has also found that in the West, there's less of a mismatch in the phenological shifts. So spring is getting earlier, leafing out is happening much more sooner. And kestrels in the West are able to predict and actually respond, basically track it, whereas American kestrels in the East have not been able to. So the next step that we want to do is actually use these genes to see if maybe the American kestrels in the East are not able to track the phenological shifts because they don't have the genetic variation to do so. which doesn't bode well for American kestrels in the east, but this is an ongoing project. But it has real world applications for what might be happening in the fitness and the population declines in American kestrels. So the third application, which is a little bit more tangible and a little bit more applied, is where we are maximizing genetic diversity of burrowing owls using captive breeding and reintroduction. And we're using genomic tools in order to do this. So this is work done by myself and a graduate student at UCLA, Kelly Barr. And one of the things that we had noticed when we were trying to create a genoscape for burrowing owls was that all of the population structure that we were seeing for burrowing owls were in the resident populations. So this up here is a structure plot where each line is an individual and each color corresponds to a probability of assignment to a genetic cluster. And what you can see is, at least for California, this is San Jose, San Diego, there's Riverside, Nevada, Arizona. All of the resident populations have pretty strong genetic, have a strong genetic signal. So they actually are their own unique, distinct genetic populations. However, all of the migrants are admixed. We cannot tell a migrant that from Oregon compared to Colorado. And this is because they're all identical and there's really no natal breeding for burrowing owls. They just move too much, at least in terms of the migrants. What was more worrisome within this analysis is that we saw that the resident populations, the ones that are actually essentially declining, are also the ones that had a greater number of individual inbred individuals. So the migrant populations, with all of their mixing, they're less inbred, whereas all of the individuals, most of the individuals in the resident populations are inbred, and that does not bode well for their population. So given these high levels of inbreeding, we wanted to ask how we can use genomic tools to support these reintroduction programs. To this end, we partnered with the Santa Clara Valley Habitat Agency, where they have they are responsible for this historic burrowing owl breeding site in the San Francisco Bay Area. In 2019, they collected 14 juvenile individuals. In 2020, they collected 13, and this past year they had collected 17. And what they do is they overwinter these juveniles and then they create breeding pairs. And that's where we come in. We come in with the genomic sequencing. We do whole genome resequencing. So again, the entire genome. And we do it with moderate coverage, and we have like 6 million variants to work at. So our goal here is different. It's not to look at population structure. It's actually to measure inbreeding. So most captive breeding programs, they will already look for relatedness amongst individuals. And if you pair unrelated individuals, that's, that will prevent inbreeding. But when you start with an inbred individual, I think it's something that we really need to look into. So with these genomic tools and these genomic resources, we can measure inbreeding on 2 levels. First, we can measure inbreeding on an individual level. So we can calculate the proportion of the genome that is homozygous, or the proportion of the genome that contains no genetic variation. And so it's identical between all burrowing owls. More importantly, and now with the advent of this technology, we can actually now map regions of homozygosity across the genome. So for example, this is just a quick cartoon. Let's say this is chromosome 5. What we can say is in these green regions, these are homozygous regions. These are regions of the genome that there's no genetic diversity, whereas it's interspersed with regions of the genome where we do see genetic diversity, where we do see variants within individuals. And with the ability to map and actually see this in space or in genomic space on some level, we can compare specific runs of the genomes between pairs in order to maximize diversity. So if we have 14 individuals and we can see this purple bird, this purple bird and this green bird have this overlapping run, we can be like, let's not pair these, because if we pair these together, these runs will be perpetuated into the next generation. And what we're trying to do is break up, and increase the genetic diversity of the offspring. And so what we end up with is this breeding pair decision tree. So the first and simplest question that we always ask, and anyone would ask when creating a breeding pair, is it the same sex? If it's yes, we stop there. And when that happens, it's an immediate stop, and only 44 of 98 pairs remain viable. Next, we do what every captive breeding program would do, is we ask whether or not there's high relatedness. Again, we're trying to reduce inbreeding. So if the answer is yes, and if it has a higher relatedness coefficient, that is also not viable. So when that happens, only 28 pairs remain viable. We're getting closer. The third and final question is we calculate individual inbreeding levels. And for this, again, this is the proportion of the genome that is homozygous, the proportion of the genome that has no variation. And there are ranges. You have individuals with low inbreeding levels, individuals with intermediate, but we had one female, and every year we have like 1 or 2 individuals that have extremely high inbreeding. So like greater than like a third of their genome has no genetic variation. And this is just a red flag for me and for anyone that would be wanting to mate or wanting to use this individual. And if at all possible, we do not use that individual. That individual will be released to the wild to create her own pair. But what we want to do is actually create pairs between individuals with low inbreeding levels and individuals with intermediate inbreeding levels. And we make the final decision to pair based on comparing these homozygous regions. So hypothetically, by doing this, we're breaking up any regions that are long and have no genetic variation. We're introducing genetic diversity into the population. And this will hopefully help with the recovery of this resident population, which has been declining in number for quite a number of years. And our end, we had 20 pairs that were good pairs and 4 pairs that are okay, the best to do in a bad situation. So we've done this for 3 years running now. And we had 5 pairs of burrowing owls that were soft released into the wild in 2020, 4 pairs that were released in 2021, and 6 pairs that are currently being released at this historic site. And while the success of these pairs, we know that they have bred successfully. What we have found interesting is the offspring has worked. We have increased the genetic diversity of the offspring when we have been able to create these pairs. So that's the end of the applications, but I just want to point out, and I think it's important, this is a very collaborative effort. And so I want to introduce you to the Bird Genoscape team. The core of it is here in Colorado, but we have our UCLA affiliates. We actually have collaborators down in Mexico from UNAM, Luz, Rafa, and Gennaro, who we had met and we are working closely with. One of the strengths of the Bird Genoscape Project, and that just goes to show like that's the size of our group and it's ever expanding, is we understand that we need to build partner networks with Latin America. This is a picture of an inaugural UC Mexis workshop held in Mexico City 3 years ago, I believe. We recently had a second one, although due to COVID, it was virtual, but we held it in the Yucatan. And this is where we make a lot of our connections in Latin America. This is essential to our work because this is where the birds are wintering. Birds are not recognizing geopolitical boundaries, they recognize ecosystems. So this collaboration across international borders is critical to success. To that end, we like to think of ourselves, connecting the dots for conservation. So it's not just connecting the dots across the annual cycle. Just picture the dots of all of those birds moving, from where they winter to where they're breeding. But it's also connecting people. So Mary Whitfield, who works in the Kern River, with Natalie, who works down in Costa Rica, collecting the Willow flycatcher. Kristen and the Bird Genoscape Project has partnered with National Geographic and Days Edge Production to actually create videos of how we do this in conservation and why it is essential that we do this. And with that, I just want to leave you, if you have not visited our website, we have this, the video is called Feathers in Flight. And it's a great introduction with better graphics than I was able to provide here of what the Bird Genoscape Project is all about. We're actually creating another one for Spanish speaking that will be shared. And with Days Edge, we've also worked with them in creating educational tools for students, for teachers, which is quite nice and expands our ability to work with citizens. With that, I want to thank, we have so many funders. These are just the ones I want to thank. But I also want to thank all the collaborators that are too many to point out in here. But the collaboration of the people, volunteers at the banding stations are what makes this feasible. Because without that, we wouldn't have the migratory links or the wintering links, even probably many of the breeding links and samples that we do have. And with that, I'll take any questions.
Fantastic. I enjoyed your presentation so much. I really liked it. I just wanted to remind people, yes. I just want to remind people, if you have questions, please put them into the Q&A. The Q&A is down at the bottom of your screen. That helps us kind of keep track of everything that's going through. And with that, Mark?
Yeah. So we have a great question from Calvin. Calvin asks, did you ever find the localized kern, Southern California, or white mountain willow flycatchers on their wintering grounds?
No, and it's unfortunate because those are also those are also likely, they're declining in numbers. I feel like kern might have like 2 or 4, breeding pairs left or something to that extent. But that's also a problem when you have such a small geographic range and such a small, it's all about numbers. Like we do as much sampling on the wintering ground as we can, but if it's that small genetically and those small numbers, we might not be able to just capture that on wintering grounds.
And Mark, there's a question.
Yep, I see there's a question in the chat. So Scott asks, your study showed that wintering southwestern willow flycatchers share the wintering grounds with other subspecies, yet remain distinct, while burrowing owls appear to mix with other populations and share their genes. Can you conclude that the willow flycatcher subspecies diverged earlier than populations of burrowing owls?
That is a good question. And no, I feel like, to me, it feels more like the burrowing owls, they just move a lot between their breeding grounds, whereas the willow flycatchers, they might be more, they return to the same geographic region. And I think that alone, so where they overwinter, that's not where they're mixing, right? They're mixing on the breeding ground. So it's all about where they're going back to. And the burrowing owls, I think they're just moving to many different places. Whereas the willow flycatchers, especially the Southwest willow flycatcher, they might share a wintering ground with like the Pacific Northwest and those eastern birds, but they only return to the Southwest for breeding purposes.
Okay, great. Okay, thank you. Lily asks, do you think the inbreeding and burrowing owls could be a result from habitat fragmentation? And if it is, do you think it could become a genetic problem for other birds?
I do. Yeah. So one of the things with burrowing owls is they are, especially the residential burrowing owls, they're in barren areas, and they're in areas that are very heavily impacted by agriculture. And so one of the things that at least the habitat agency that we're working with, they're providing also artificial burrows. The burrowing owls also, they don't create their own burrows. They take it from, prairie dogs or something in the area. So even the movement or extirpation of those burrowing owls, if they don't have places to breed, they won't, you know. So I think, yes, I definitely think that it's always an issue, habitat fragmentation and lack of, you know, optimal breeding areas is going to be an issue. One of the interesting things, and this is just a side tangent for burrowing owls, is in the Imperial Valley, there is actually an increase in agriculture, an increase in, but interestingly, it's actually helping them on some level. What they're seeing is actually a great increase in burrowing owl numbers there. And what they're seeing is actually migrants that overwinter in this imperial area. They're not migrating again. They are actually staying and breeding because there's this pulse of resources from agriculture. And so somehow that's working in their favor, but that is super tangential, and we don't have like real data to support that, but it's interesting on so many levels.
Cool.
That's interesting. Do you know, Kristen, how your data is being used by ornithologists as far as creation or support for subspecies?
No, not for the willow flycatcher. For burrowing owls, we've been talking with like a lot of groups in terms of like translocations, right? Like, should we be moving birds from San Diego up to, you know, San Jose? And we're very tentative or hesitant to say yes or no. I mean, the fact that they're distinct genetic units is, to me, points to maybe you shouldn't be moving them.
Oh, right.
But they're also declining populations, right? So like maybe an influx of different genetic material might help, but you don't want to mess up like local adaptation at that scale. So it's one of these things where we can, especially for management, I think it helps to think of them as different populations and distinct. It's just, we don't, I don't take that extra step to be like, yes, let's define this as a subspecies or something.
Right.
Yeah. For me, I'm just like, it's different. We should maybe prioritize this. It's unique. Let's preserve as much uniqueness as we can.
So I have a related question about the willow flycatcher. If you say quantify the differences between some of these, you know, new populations, the Kern population, White Mountain, and the Southern California one, are they quantitatively different enough that you would expect that someone, you know, that you know, eventually they would be recognized as subspecies, or could be recognized as subspecies, or are the differences, you know, detectable by you but smaller than the differences between the other subspecies?
I think that's always going to be one of those issues. Is what do we consider quantifiable? What's the difference? You know, so I know birders, it might be, so in fish and salmon, migratory timing is one region on the genome, one gene. Is that enough to make this, you know, a different status for like spring run or fall run timing? I think that's always going to be an argument, but I think understanding that there is a difference and that has like phenotypic and like behavioral differences in terms of like where they're breeding or like if they're mixing, I think it's important to note. But I always, that question is like, is it enough to consider it different? I'm like, it's different, but I don't know what is enough.
Yeah, no.
Yeah.
Yeah, I guess what I was asking more, are the differences, you know, as large or larger or much smaller than the differences between, say, the, you know, the Pacific willow flycatcher and the Southwestern, for instance?
Yeah.
Or are they just different, you know? Yeah,
it's true. So when we create our primers, we try to represent the, you know, the structure that we see, but we're going from millions of variants down to like 96. We try to make like a panel of 96. That Pacific Northwest might only need 4 variants because it's so different, whereas we might need like 10 or 20 in order to get that that structure that we saw with the with the kern, because it exists, but it's just there was less differentiation between them. Okay. So it's very dependent on species, and it's definitely, yeah.
And what markers you use and so
exactly.
on.
Which ones you choose. Yeah.
Okay. All right, cool. We have another question from Marie. Wouldn't the migrating owls that stick around improve the genetic pool, or do they only breed with other migrants that also choose to stick around?
So that's a good, they actually do breed with the residents. So what we're seeing in these switcher populations is they look more like migrants, like that. What was potentially a resident and distinct, genetic unit is now, falling out in terms of like genetic space with the migrants. Is that helping? Potentially, but I think we still need like the actual fitness, like in the field. How are these birds doing? How's their, what's their clutch size? What's their, everything? Like we just need to see how they're doing to see like, and have actually comparison between these resident populations and these switchers, just because if it is helping, it's definitely pretty, it's an interesting thought that we could, have that happen.
And now I know what a switcher is.
Yes.
I thought, well, never mind.
That term might not catch on. That was just for this presentation. Yeah.
Great. Let's see.
And I think we are out of questions. Well, Christen, thank you very much. We had a lot of compliments in the chat on the wonderful webinar and presentation, and thank you very, very much. And I just want to remind everyone, next Tuesday at 7 o'clock, we will have Adam Searcy here, who will help us to differentiate between swallows.
Swallows.
Oh, yes. And so it's a great migration. If you haven't been up to Bear Divide, please go up to Bear Divide. You will have a wonderful time up there and learn something.
And go on to, before you go on to Bear Divide, go on to Birdcast, which will give you the reasonable probability whether it'll be a good or not so good day at Bear Divide. Ryan tells me that Birdcast has been predicting the good days pretty well.
Good. And if you can't actually make it up to Bear Divide, their website does have a live feed as to what they're actually counting and seeing. So that's really exciting to watch. And I think that's it. Thank you again, Christen.
Thank you so much, Christen.
It was
wonderful.
Thanks for inviting me. This was wonderful.
Oh, thank you. And we will see everyone next week. And until then, good night and good birding.
Good night.
Good night.